All right, so the problem we are considering here is the following. So imagine that we have a population that happily lives in its original environment, but things can't go on smooth forever. So at some point we have an environmental change, a severe environmental change, and the population becomes maladapted. And like if nothing else would happen, would then be on its way to extinction. And now the question arises, of course, right? So can the population escape extinction through adaptive evolution? And so this is the key question that's, all about this thing of evolutionary rescue. And we've started to work on these questions a few years ago, and this has been done together with Hildegard Uecker. Actually, this is part of Hildegard's PhD thesis, and also in collaboration with Sally Otto from British Columbia. And most of the results are actually have just appeared in the American Naturalist in January this year. All right, so what's the empirical interest in these questions? And traditionally, this comes from two different sides. On the one hand, we have, conservation genetics, and usually there people believe that questions like evolutionary rescue are really important. But one must say that the data they have is, not so overly convincing as yet. And the primary reason for that is that it's just difficult to make the case of evolutionary rescue in natural populations. Imagine there are very many cases where we see populations that do this adaptive tracking of environmental changes, right? So, and this has been well documented. And there are many other cases of populations who don't succeed to do these adaptive tracking, and then just go extinct, either locally or globally. But for this evolutionary rescue, we would have to make the case that the populations that were able to track a changing environment would have gone extinct otherwise. And this usually is very, very difficult. Also considering that, of many of these ecological data, where we have good data for population counts and so on, right? We basically lack precise genetic information. Now, whereas here in this first case, of course, right, so evolutionary rescue is something that we may want to see. In the second case, it's just the opposite. There it's, rescue is the same as evolution of resistance against antibiotics or pesticides. And this is usually a case that's much easier to make, simply because human comes with lethal selective pressures, and we do know that populations would go extinct without these bugs to adapting, and frequently, much more frequently than we may hope that they actually do. But so as of recently, we still have a third source, right? So where we can hope for data, and some new data is being generated. And this is from experimental evolution. And of course, this is always the field that the theoreticians love. Some of you have already also started to do experiments on the side of there being theoreticians. And so this is like a very nice combination. And so in this case of evolutionary rescue, the main experimental work has been done by Graham Bell and Gonzalez. And this is their setup. This is like yeast that they've plated here in an array. And then they have like a salt concentration that increases like a gradient from like a very low concentration. So this would be like the natural environment for these for yeast, and then increasing more and more just in this direction. And then what they do is just to increase the slope or increase this concentration gradually across this array more and more. And at the same time, right, I mean, with the help of a robot, obviously, right? They do local and global migration, or global migration among these plates. And then, in the end, when the total population then arrives at a salt concentration that is lethal for the wild type, then they just record whether there has been evolution of rescue.
Sorry, they're starting with high concentration at the top and low at the bottom, and then gradually increasing it?
Exactly.
Everywhere else?
Yes, exactly. Yeah. Which, for a first experiment on this, is something that is already moderately complicated. Indeed, a bit too complicated than easy theory that we can do would directly apply to these experiments.
I guess if they just had the gradient from the beginning and just kept that gradient, why this way? Or is it?
Yeah, I mean, this is what they did, right? So it's this is what I would have done as my third or fourth experiment, not the first one, but I mean. Yeah, don't know. All right, so this is what we have as our empirical basis. So what does the theory say? I mean, one basic thing, of course, is, the complicated thing, or maybe also the thing that makes it interesting, is that evolution and ecology cannot be separated for this problem. I mean, we have a declining population. This is this race of demography versus evolution. So necessarily, these two things happen on the same time scales. Amazingly little work on precisely this situation, where we really have population decline to extinction being possible in a model, has been done until quite recently. I mean, there has been work with a quantitative trait, basically something with a fixed variance, and then we see that if this variance that we have fixed in the population for this quantitative trait is large enough to be able to cope with an environmental change at a certain rate by Holt and Gomer-Kiewicz. And some work in permictic populations, mainly by Alan Orr and Unclis. But then, recently this seems to be now a new emerging topic. There has been a special issue in the proceedings B right on this topic. And people have started to ask, so can we understand evolutionary rescue under more realistic ecological conditions? Because, this is something that reacts to an ecological selection pressure, obviously, so we should have some moderately realistic ecology in our models. All right, so, and this is also our starting point, of course. What we did is to consider population structure. And the reasoning behind this is as follows. Environmental changes will often be rather gradual than sudden, and in very many cases will also not affect the total species range at once. And this is even true for antibiotics or medication in the human body. It's not all parts of the body are affected in the same way or at the same speed by the antibiotics than others or other kinds of medication, and is, of course, always true out there in nature where populations inhabit complex habitats. So, since this is really the first model that's done here, all we want to do is to explore what's possible. What kind of effects could you expect to see if you have this combination of factors in the model, space, declining population size, and so on. For example, what is the role of temporal refugia, right? So if you have not the total range degrading at once, then you have the possibility that, the wild type still holds out in some areas of the space and sends migrants, and maybe these adapt and so on, right? And also, what's the impact of the degree of fragmentation of a habitat? Does this make it easier or more difficult for populations to adapt, really? And also consider some other factors that we do have in our model. This old question of standing genetic variation versus new mutations. So does rescue come from variation that's already there in the base population when the new selection pressure starts, or does it come from new mutations that only arise later on? And also, what's the influence of competition maybe that we may have between wild types and mutants? And also, this will be an important point. All right, then I should come to our model. And so, since this is the first model, we just did the easiest thing we could think of, and yes, to use an island model here, right? Where we have D islands here that exchange migrants with a migrant pool at a constant rate of m. So all send out migrants and receive migrants from this one common migrant pool. Then we have like just two genotypes, the wild type and the mutant, that are connected by a mutation rate of u. I mean, so this makes things really simple here, only two genotypes. And then, under the original conditions, what we assume is, I mean, that the population sizes on all these islands are equal and constant and given by k. There's a wild type which has a fitness of 1 here, and mutants have a fitness of 1 minus z, which means that they would segregate here in mutation selection balance to form the standing genetic variation in the base population. So z can, of course, also be 1. In this case, we don't have any standing genetic variation, right? All right, then, I mean, the environment starts to change, and we model this like this, that every t generations, one of these demes switches from good to bad. And under these perturbed conditions, then we have the following fitness scheme, right? So the wild type here has an absolute fitness of 1 minus r. So that means it would just like within this deme exponentially decline, plus of course as long as there still is wild type in these other demes, there is like immigration from these others. So we have decline plus immigration. The population size on the kth island, like for long time intervals t, then would approach a migration selection balance. For the mutant, we have the following scheme here. So this is the absolute fitness given by this mildly complicated thing, but I mean, first just consider this 1 plus s, so which basically means the mutant can grow under these new conditions, right? If s is larger than 1, which we assume, right? And then there is a beta term that models the density dependence, like so the degree of competition that this mutant experiences from the wild type in these new demes. Like if b is large, then we have a strong density dependence. This is the total population size in the kth deme here, right? And if if beta is small, then I mean there's almost no density dependence. All right, so after a while, of course, then all these demes have fully degraded, and now the wild type certainly will decline to extinction under these conditions. And now the question arises, can the mutant establish to rescue the population or not? And we generally have a rescue probability that depends on all these model parameters here, right? So it can be quite complicated. But actually, I mean, for this talk and also mostly for what we did in the paper, we focus on two parameters of particular interest. And one of these is here, this R. And R, if you remember, this is, I mean, measures the strength of the environmental change. This is the rate of decay under the exponential decay of the wild type under these new conditions, right? And here, obviously, you have this question. You may think for yourself what you expect. Will this rescue probability rather increase or decrease if we have an increasing strength here of the environmental change? Of course, if you ask an average ecologist, so this would be the expectation that this should go down. And indeed, this is also what's confirmed in this experiment of Bell and Gonzalez. In this one case, of course, I mean, they only had 2 data points, right? So that show the decline with a with a faster, like if you do this increase in the slope, what they did faster or slower, then they had more rescue in the slower case. Okay, the other parameter that is of interest to us is the migration rate. So again, you could think of what you would expect. Does it rather go down or up? The rescue probability. Again, most ecologists seem to think that gene flow is a good thing, or alternatively said, fragmentation is a bad thing, or always fragmentation is a bad thing for ecologists, right? And therefore, if there's more gene flow, then it should go up again and again by 2 data points. This is confirmed by the experiment of Bell and Gonzalez. Okay, let's now come to the analytical approach that we have here. Okay, so the model for doing analytics is, like in its original form, too complex, at least for us. So what we do is to simplify it a bit. And the first thing that we do is that without density dependence, right, so we really can distinguish just like 2 compartments, right, so in this model. This is the part of the habitat under the original conditions and the part of the habitat under perturbed conditions here, right? And then after each time step, this will shrink and this will grow. And we also have here a time-dependent migration rate among these 2 compartments. Then. Now, right, okay, so in this case, we still have to consider the following. So we have a wild type, right, this is an O, in the original part of the habitat as a function of time, right? This then here exchanges migrants with a wild type population in the perturbed part of the habitat, and here we have mutation, here we have also mutation, and here, so here's a mutant, mutant original, and a mutant in the perturbed conditions. Again, we have here migration in both directions. So how do we approach something like this? So, I mean, luckily, we only have mutation in one direction, so we can separate the wild type part first and do this this part first, sort of, and we will try to do this part deterministically, just like with a differential equation. And in the end, of course, I mean, we are mainly like here interested in this part. This is the mutant under the perturbed conditions, right? So these are the rescue guys, right? And we will want to do this by a branching process. All right, so this is the idea. But I mean, as you see, still in this scenario, we have here migrants from here to there, and also everything, like so the inflow from the wild type and the migration rates, these will be time dependent. So effectively, mathematically, this is a problem of a multi-type, like a 2-type, time-dependent branching process, right? And yeah, as it turns out, at least for us, this is still something that's too complicated to get any explicit results, right? And therefore, we still need to simplify the problem a bit further. And the first idea here is, I mean, what we do, that, I mean, we ignore this arrow, right? We say that, migration doesn't really take us to these original conditions, but we do as if, I mean, everybody who migrates out again from these perturbed conditions just dies, right? Basically assuming that offspring that once went here will never come back here to these conditions, which will be a good approximation of the fitness here of the mutants is not so good, right? All right, so this is the first idea, and then we will see we basically come down to a single type time-dependent branching process, and then we are in the game. Okay, so as I said, this is the idea. No back migration of mutants, right? So this is the first sub-case that we consider. And this is the wild-type dynamics. So this is a differential equation that we somehow can do. Depending on time steps and exponential decay, we get some step thing out of this or whatever, but this is doable, right? And then here, for the mutants under the original condition, so this is this edge here, so we just also model this deterministically. This is a deterministic mutation selection balance, and now we come to the interesting thing. This is the branching process in the perturbed demes here, where we have like a constant birth rate, right? But here also like a death rate. There's just 1 plus all the guys that really migrate to the original demes. This d minus small d of t, this is the part of the habitat that is under the original conditions, right? Okay, so we effectively then get a selection coefficient here, just like this, something time dependent, as I said, and basically we can fit this in for any single mutation to get an establishment probability. There are, like, say, ready-made formulas to fit in these selection coefficients, and we use those here, right, to derive an establishment probability for each single mutant under these perturbed conditions. Once we have this, then it's easy to get a rescue probability. So this is just done like a Poisson probability here. This exponent would be the probability that none of those, rescues it, and this 1 minus the exponent then is the rescue probability. If this is the total rate at which rescue mutations appear. So this n rescue here is this thing, right? And this for this we have 3 different components that contribute to it. One is this comes the new mutations from the wild type in the perturbed demes, right? So this is this transition here, right? Then we have the migrating mutants from the unperturbed demes, so this is this direction here. And all these are multiplied by the establishment probability, of course, and then also we have like another contribution, and this is what's called pre-existing mutants at environmental change. So every time such an island switches from good to bad, we still have some mutations sitting there, and we also need to consider these, right? All right, okay, so, something will come out of this. I will come back to this in my results, definitely, right? But okay, there's a second case that we actually can do, so let me first mention this. And this is the case if we have migration rate of 1, which in our model just means that everybody migrates back to the migrant pool, and then it's just redistributed to all the demes in the end again, right? And this, again, makes things easy. We can do the wild type thing, no problem. But like the essential thing is that we now can do the branching process here in the migrant pool. Every generation, everybody will come back to the migrant pool. So this is a single compartment. This, again, will be a single type time-dependent branching process. Actually, if we do this, we get like an effective selection coefficient in the migrant pool for the mutants that reads like this. And you don't have to understand all the details, just note that here's an R appearing here. So this is a parameter of the decline parameter of the wild type under these perturbed conditions, right? So it's not a mutant parameter, it's a wild type parameter. And it just says that, of course, if the wild type heavily declines here, it will send fewer guys back to the migrant pool. and therefore competition of mutants and wild types in the migrant pool is more biased towards the mutants in this case, and therefore we have this in the selection coefficient of the migrants is also affected by this R. All right, and again we get the establishment probability from the formula before and the rescue probability later on. All right, last case that we also can do, right? Or first, we should say, until now we have ignored density-dependent selection here, and we also wanted to do this. Now, if we include density-dependent selection, then we are in trouble, and this is because then, the actual numbers of wild types in each of these stupid islands really doesn't matter, we can't use this two-compartment thing that we have done anymore. And therefore, I mean, all we could do is to make things really easy and do like a dramatic different condition that we just put a migration rate to 0 in this case. So then we have independent islands really. And of course, we then do these branching processes on these single islands. But I mean, it turns out that this is indeed an interesting limiting case because it does not only describe the case m equals 0, but also, of course, like if we have just a single deme, easy to see. And also, if we have a sudden change, so that like if t is 0 and all the demes change at the same time, all these three limiting cases are described by the same thing. So, again, something that we can do. But now let's come to results, right? And again, right here as a function of the severity of change, you can think again what you expect. So this is what we do see if we have no density dependence in our model. So like a decline with the severity just like expected for either high or low standing genetic variation. But if we have density dependence in our model, then we see the following, and in particular, of course, this is here notable that with high standing genetic variation and density dependent selection, we get this, first a decline, but then it increases again. All right, let's look at this particular case in more detail. So this is the limiting case that we can make with no migration. So the lines are always then like analytical results. The dots are simulations. I mean, always, if we can make it, can do it, I mean, this branching process approximation is excellent, right? And okay, what we can do here is to distinguish the contributions of these two sources of of mutants. New mutations here. This is this lower line, and pre-existing standing genetic variation, this is here. And there we really see that this increase is done by pre-existing standing genetic variation. So standing genetic variation is responsible for this increase here. Again, the question, why really do we see this increase? How can we understand this intuitively? That's not so difficult after all. It's interaction of standing variation and density dependence. Standing genetic variation is the main source for mutant alleles. And then we have density dependence. Adaptation is only possible with low wild type densities, and therefore like a fast decline of the wild type is needed because otherwise all this standing genetic variation is lost before it can get effective, right? So before it can really grow. And this explains why in this case it can be easier to adapt to more severe changes. But then we also have this case, right? And this is the Levine model, meaning migration rate of 1 here. And note that in this case we did not put in any density dependent selection in the new demes. But nevertheless we do get this characteristic shape. Of course, I mean, if the severity goes to 0, then at some point also the rescue probability must go to 1. This is trivial. It goes down first, but then we have this marked increase here again. And so the question arises, why do we see this increase in this case even without density-dependent selection? Now, of course, I mean, the reason that we've already seen here is this R dependence of the effective selection coefficient here, right? So that in the common migrant pool, we do have competition among wild types and mutants still, and this is what drives this increase here again. So relaxed competition, because of a lot of wild types dying here, can be easier to adapt to more severe changes. Okay, let's now get to the second model parameter here. So this is migration rate. Again, you can think of what you expect. So this is what we get without standing genetic variation. And now you can think of what would standing genetic variation change in this case. Now what it does is this, right? So get quite complicated patterns in the very non-monotonic shape as dependent here on the migration rate. That's a log scale here. First question, why this intermediate maximum here? So this is easy to understand actually. Consequences of migration on the dynamics in the perturbed demes, like here on the migrants here. So we have two conflicting contributions here. First, wild-type migration is usually beneficial because, I mean, of course, like increases the wild-type population size here in the perturbed demes, and there's more mutational inflow here from here to there that could happen if we have a larger source here, right? So, and this is basically a gain that is proportional to M somehow, and then on the other hand, of course, the mutant migration, just in this case, this is most obvious, right? If the migrants here just die, right? This is detrimental from perturbed to original, and this corresponds just to a loss of mutants, and we roughly get an increase in this. The increase of the mutant population in the perturbed conditions is just proportional to something like S minus M. And this explains this decrease a little later on again. But then, okay, we have this standing genetic variation, and then we see that we get this secondary increase here, and we need an explanation for this. I'll try to give this. And this actually is because of consequences in the dynamics, not in the demes under the perturbed conditions, but in the demes under the original conditions on that side, right? And these become important if we have standing genetic variation, and therefore a lot of mutants also in these demes, right? The wild type, if we consider this just faces some loss due to emigration, and we almost don't have any gain due to immigration because it's so strongly hammered in these new conditions, right? The mutant has loss and gain. So for mutants, this is just some is about equal, I mean, these two sides. And this leads to a relaxed competition for mutants in the original demes, right? So even if we do not have any explicit density dependence here, just because of keeping population sizes finite under the original conditions, we necessarily have competition of mutants and wild types, of course, because of finite population sizes here, and this is enough to explain this effect, right? And then, like this leads to migration selection balance at about M over Z here in these original demes, and with this we can then now explain the full pattern. Here, effects of migration with standing genetic variation. We have wild-type migration will lead first to a gain proportional to M. Then we have a mutant migration, reduced growth in the perturbed demes of if M becomes of the order of S. This is the S here, right? And then, like, if the mutant growth in the original demes is possible if M becomes larger of the order of Z, actually, right? And here, so the Z in this particular case, and this explains this secondary increase here. All right, so with this I'm already then at my summary. Yeah, evolutionary rescue, ecology and evolution intertwined, and this necessarily always in these models gives us a large number of factors that we need to include. And our results really show that it's necessary to include, a large number of factors, because these strange effects only arise from a combination of different factors and give sometimes unexpected behavior. And the first thing that we've seen is that rescue for severe change can be easier than for moderate change, either due to competition of wild-type and mutants in the new environment or due to competition of both guys in the old environment, like if migration is strong. And when really broadly, should we expect this effect, right? We get it regularly in almost all cases in our model if we have strong density-dependent competition and a lot of standing genetic variation. So these two factors together really drive this effect, right? And now we can, of course, go back and ask, has something like this ever been observed in data? And indeed, there is one case where people have pointed this out, this PNAS paper of Reed and al. On malaria. So they see like a fast eradication of drug-sensitive malaria strains, and this allows for a quick amplification of resistant types from standing genetic variation, faster than in cases where this eradication has been slow. Right? So this is directly confirms to what we see here. All right, second thing is then migration. And there we simply have seen that there are a number of ways how migration can affect this rescue thing, right? 4 different ways. Actually, right, migrants, wild-type migration increases source of new mutations, mutant migration decreases establishment probability. These are the first 2 effects. Then with density dependence or competition, we get 2 more. Actually, I've only shown 1 here. There is one other that I've not shown in a graph here, that wild-type immigration increases competition, of course, and wild-type emigration decreases competition. The last one is the one of the Levine model that we have seen. And generally, we would then expect a non-monotonic dependence of rescue on gene flow. All right, so, but then if you just want to keep a single line as take-home message from this talk, maybe this is the most important thing, that the presence of the wild type can be bad for evolutionary rescue, right? And I just, with a few words, I just want to make this point that this is even generally true than just under the scenario that we've seen here, which has been competition of wild type and mutants, right? So certainly one important factor. But like ongoing work really also shows that we do not even need really competition. It also works with the recombination. And now, what we started to do is that we think of situations where rescue is not only possible to a single mutation, but you need 2 to get rescue. 2 drugs, for example, right? That's also called complex rescue due to a double mutant, right? And then we see this dependence of the rescue probability on the recombination rate here. Again, this is an intermediate optimum, maybe of interest also to people like Dan Weissman, right? So even without tunneling, we get some intermediate optimum here. But what I want to point out is this thing. Like, this is the case for a mild effect on the wild type, and this is the case of a severe effect on the wild type, right? In this case, the problem that the wild type causes, it's just that it makes recombination of the double mutant and the wild type, so much more likely, and therefore, so it decreases, presence of the wild type decreases, therefore, the rescue probability. But this is work in progress. Again, that's work done in collaboration with Hildegard Uecker. Should really point out that, the bulk of this work has been done by Hildegard. And with this, I'm really at my end. Thanks for your attention.
Confirm these are a waste of time.
Yeah.
Just ignore them. Are there any questions for us?
Yeah, that was really interesting. I'm wondering, if you go back to Bell and Gonzalez experiment, experiments, though,
Yeah.
and I know that they were somewhat more complicated than just the ideal, so they don't really match your analytical system
Yeah, yeah.
perfectly. But I'm wondering, because there they found, as you mentioned, that faster degradation of the environment, so then higher R, or sorry, not faster degradation, a stronger degradation of the environment, so higher R, would actually result in lower probability of rescue as
Yes,
expected,
yes,
right?
yes.
So I'm wondering, I mean, given that these youth populations that are rescued is likely to be from the same genetic variation, given kind of the low mutation rates, right, relatively low mutation rates, and presumably there's quite a bit of competition between these yeast strains. So why do you think they actually found this effect when your work seems to indicate that that you might actually have an opposite effect because of these density-dependent interactions?
Yeah, I mean, of course, there are several possibilities. I mean, first thing, of course, is that even like in our graphs, right, we first see a decline, right, and then a secondary increase again, right? So it's a non-monotonic thing, right? And if you just to try to do this just with 2 data points, right, something non-monotonic, right, so it's maybe just not enough, right? So no idea, right? So even if you just do more than this, I mean, this could change, right? And then of course, I mean, yeah, they certainly did have rather low migration rates, right? And it depends then, right, whether really like competition is strong enough to inhibit mutant growth for a long time, right? And this I also don't know, right? So this is the second possibility that I see, right? Just one here and then here.
So it's obviously very cool, but I found myself a little confused by the idea of having absolute fitness in some beams and relative fitness in other beams. And I wonder if there's any easy way to sort of make it more regular, and if even the one with the 3 humps might possibly be an artifact of that in some way.
I mean, you can start the model with absolute fitness in both beams, and on top of that, have like a population regulation in the odd beams. And this comes down to the same things. We even have done some simulations where then it is, I mean, possible. I mean, then it's a stochastic effect that depending on what average number of offspring you have, but not in all cases, then really, it's like if you're really unlucky, then you also don't fill these old beams quite fully, right? But I mean, usually they are just filled by 95 to 100% or so, right? And at that point, there's just like an external population regulation that is just affects mutants and wild types in the same way, right? And then to turn this into a model, right? It's just much easier to assume relative fitnesses only, like in the old beams, right? But it does describe a scenario where you have absolute fitnesses on both sides. And then again, of course, in the new deems, there you do need the absolute fitnesses, because rescue and everything, this does depend only on the absolute fitnesses. Right?
I guess I'm just wondering if you should do all your simulations with absolute fitnesses and yet
We
keep the
did-
model the same.
We
since you think it
did
should be
some
better.
of those actually, and we didn't see a difference, and then we changed it, and because it simplified things, right? To keep this population size as a really constant k, then
Do you expect the spatial arrangement of the deems to make a difference? For example, if they are two or one dimension? I mean, here.
Yeah, sure. I mean, this is like a step that one of the steps we would have to go to get closer to the Bell and Gonzalez thing, right? Since they also did this stepping stone thing, right? Yeah, so. We thought about this for quite a while, because this was one of the lines we were thinking of going on. And in the end, we came to this idea that it probably will really only make a major change if we have this multiple step rescue thing, right? So if we need more than a single mutation, and then we thought, okay, we should do multiple steps as such first, right? Before, don't know when we will get to these complicated things. Yeah. Of course, there are some changes, but I don't expect that the qualitative patterns that I've shown will really change just with stepping stone and single point, single rescue mutation. Was here.
So I was similar line, like what is really the basic difference between your type of evolutionary rescue models compared to especially structured models, where we don't have the change in the environment, but we just have a structured heterogeneous environment. And related to that, some of the patterns you seem to describe or find in models of evolution of plasticity, where we basically have initially plasticity as a somewhat the rescue, and then you have basically the tracking via fixed genetics in changing environments across time, basically provides similar patterns that what you find. So for that, basically the time and space is somewhat the same.
I mean, yeah, so even more closely related models called models of niche expansion, right? That you have like a wild-type population that sits in one deme and tries to colonize like an island where the wild-type can't live, but maybe mutants can live, right? And indeed, you do see some of the same effects there, like this density-dependent effect has been described there in a model also by Gomolkiewicz, right? You do see this sometimes, but the essential difference always is that there's no ecological timescale there, right? There's never this race. I mean, they always will have this, these niches being colonized, like then at one point, if it's at all possible, right? And this additional timescale that you have, like say, you need to be quick, right? This, has rarely been considered there, right? And also like makes an additional aspect definitely about this rescue models here.
Okay, we're running very short on time, so one very quick.
Well, I just want to go back to the ecology of original organization relative to conclusions. It seems to me that especially in this sort of considered conservation, it's not clear that at the near extinction in local populations, there's a monogenicity with density. And the fitnesses very often go down very much quicker as the density gets close to zero for many organisms of interest in conservation biology. And then secondly, and not to mention the fact that there are quite a few resistance systems where there's some cooperativity that has to do with establishment. So it's not a single, it doesn't have density, simple density either. And then finally, most of the pesticide resistance very often involves diploid genotypic differences in fitness. So for example, typical insecticide resistance will show a heterozygote will be more fit, but actually the homozygote is less fit in the selected environment.
Oh, definitely. I mean,
And
so this is,
all I'm saying is that leads to very different dynamics without any density.
I mean, this is absolutely acknowledged. I mean, it's just two quick comments maybe. We see our models definitely much closer to this, the resistance thing, which are in many cases just driven by few loci, right? Then to, conservation things where you have really quantitative traits and very many genes are involved, right? And also there's not such good data. Second thing, of course, this is the first try, to try to explore this territory. There's much more to be done, right? So, dominance effects, for example, would be one thing.
Okay, well, you've got this afternoon to
Yep.
solve all that. And I think we're going to move on.